Good morning, everybody. Thank you for inviting me to this forum. And today I would like to share a short story which happened over this summer. We decided that it's about 3 years since allotrope been in existence, and it's time to test the practicality of the promises they gave to us and test how universal is a problem being addressed. So we decided to test certain things. What resources do we need if we go into start a pilot in the production? And what it takes in terms of personnel, how to get there. But before getting in it, let me describe the subject and maybe introduce a little bit about myself, where we're coming from. So how do I press this? Oh, yeah. So I'll briefly talk a little bit about complexity of and the challenges of projects, how complex data we wanted to move in ADF format. Maybe describe a little bit what constitutes our data. Talk about our endeavor with Thermo Fisher file format, which being a robust metabolomics prompt platform for us for many years. And then talk about issues and storage ideas, and maybe finish up with a little challenge. We are ready to provide, to donate to allotrope some data to go and try to solve that problem maybe more efficiently than we did. So I'm representing here Apply an Investigational Metabolomics team, which exists with BMS Pharmaceutical Research Institute in Preclinical Drug Optimization Division on the Bioanalytical and Discovery Analytical Sciences Department. So metabolomics is quite a new science. It come into existence and maybe start being in industry in early 2000s. But it's from the start, it had a lot of promises. Now promises over those decades has turned in solid deliverables. And what we can deliver now with metabolomics tool sets is address some mechanisms of both toxicity and efficacy of pro-drug candidate, look at biomarkers, maybe identify and stratify our patients in our trials or samples in our studies, and try to give some real-life information to build models, try to get to a system biology. And of course, it's very clearly that metabolomics can touch any part of drug discovery process in pharmaceutical industry with very early to very late stages. And it's not limited only to biological samples. It could be used in optimization of certain chemical processes and many other things. So I can go on forever about metabolomics and what we do and how we do it, but I decided to compress about 150 slides in one slide and said there is a bottom line. There are two major approaches in metabolomics. One is non-targeted, which in early days was known as a fishing expedition, where you try to enroll analytical methods and measure everything you can, and then try to make sense of what you have measured. And another approach is targeted approach, where you exactly know what you're after, what molecules you're after, and then try to build and optimize your methods to precisely measure those molecules. And of course, when you try to do a targeted approach, you're more heavy on method development, but it's easy on data interpretation. When you go to untargeted approach, you maybe have the same time of method development, but interpretation and data mining afterwards is a big time-consuming process and still a challenge. However, if you look at both processes, data collections is fairly automated process. After you've done most of the time you spend in your lab to think about how you prepare your samples and how you introduce them into your measurement instruments. But when you've done that, you have plates, you have robotic autosamplers, you put it in autosamplers, press the buttons, and almost every vendor who's sitting in that room knows that they can produce very compressed, very nicely built and written files packed with a lot of data. So what we decided early in the game, how about we can take best of the both worlds and collect data in a targeted manner, but to make our bosses happy and deliver on promises on a turnaround time, we can mine those data in a targeted fashion. So we will go only after those signals which we know how to interpret and explain. Among all techniques we have at BMS and employ in metabolomics, and it's a classical mixture of different separation techniques, usually backed by mass spec detector, but we also have NMR. There are also possibility LC-NMR, but it didn't meet the robustness test, so I'm not showing it on the slide. In this short process, we decided, well, we are not ready to consume data from all of that instrument, so let's focus only on high-resolution LC-MS. So it's try to limit the scope of the process. So typical bioanalytical workflow, and I'll get to a specific workflow. What we have at the start, we have some study metadata where samples are prepared, introduced into some sort of instrumentation, raw spectra collected. So in one bin, you have metadata and your collected data. Next step, you apply some automatic pre-processing, maybe some sort of alignment because you want to compare across your samples, and then maybe apply some sort of criteria of the quality of what you have collected of the thing, and then filter and do statistics. So that would go into analysis then, analysis and algorithms. And finally, there is almost like an open-end question. Okay, now I have my measurements, which I have assigned identity. How do I put it into biological content? So obviously there is a big chunk of work which I call interpretation procedures. It's very difficult to define them because usually they're in the head of the scientist who is doing that procedure. And if you ask it, you know, today I'm doing this, tomorrow I'm using CAC. Yes, day after tomorrow I'll be using HMDB, a human metabolome database or something like that. Maybe I'll employ MATLAB and try to do system biology in there and put my measurements into that procedure. Now, we decided to limit the scope and say we limit to one standard screen, which we have been running for years, and we have lots of examples on both proprietary and non-proprietary systems, so we can work with academics. And part of this project was where to get resources. And we decided to, we had ongoing collaboration with Rowan University through our innovation grant. Specifically, we were working with a computer science students and students from bioinformatics department. So we decided, well, this is good. If we limit the scope a little bit, we can see and use them as a guinea pigs and subject them to this plethora of allotrope training materials and information and see whether they can survive, and we don't have to enroll them in a specific medical institution afterwards. So workflow was limited that we looked at typically 6 groups and 6 samples. Also, you have to run blanks and QCs with this. That's a standard robotized procedure with protein precipitation, which yields you a nicely formatted 96 wall plate, which is goes into autosampler. And this is where it gets interested. In maybe a runtime of a couple days, it's on order 2-3 days, you are generating a lot of data, and typically it's about 800 gigs of data being generated in such short period of time. Then data are subjected to process, and obviously you fill out some sort of report. And if you just look at that report would say, we have detected differences or metabolic differences between our control group and dose group or some sort of disturbed group. it's immediately become understandable that everybody through presenters through last 2 days at Allotrope saying Allotrope is great at this, and we are preaching to the choir. So yes, we are the choir. We agree with you. It's time now to take that information and leverage and mine it and put it into content. So everything what we have done in 10 years could be leveraged and see, okay, I detected this metabolomic signature where I have seen it before. Did that drug candidate proceed into a next step, and how? So it's become very practical question. And obviously, what happens next? This is a typical workflow, and I try to map that workflow in our lab in days of turnaround. So early on in the game, our toxicology colleagues decided that you have 2 weeks to deliver results. So obviously, I'm not counting planning stage when the study was designed, from times when the samples were delivered to the lab to the time you give a report is approximately 2 weeks. So I try to map what we're getting in those 2 weeks in 7 days. So from the moment you receive the samples, what you try to do, you try to scavenge all available metadata, do some sample preparation, and build this nice 96 flow plate, nicely formatted and ready to inject into the instrument. But you want to retain 2 things with it. Everything what happened during the sample prep is the record because it's useful for troubleshooting. And the other thing, you obviously need some control file which you send farther down the road to the instrument so you know what injects in which order and position. Obviously, this is now we're getting into slightly off topic because this is the statistician realm which will tell you that what injected from the edges of the plate is not the same what's inside the plate and other interesting sampling theory questions. Then from day 3 to day 8, this is an exponential growth of data. This is where lots of raw data is being collected, and we are not waiting for them to finish. We can start pre-processing them as they go. And then rest of the time is spent on the signal processing, annotation, interpretation, doing statistical analysis, putting in biological content, and hopefully it ends up in some sort of presentation. It's either PowerPoint time or more formal Word document, PDF file, which is sent to working group to review and make conclusions. So in 2007, I also, if you notice on my left side, there is a logarithmic scale in terms of file sizes. So the way how we organize it is a network file share. As the process proceeds, we deposit files in a specifically designated folder. So I can track how it goes with time. So in 2007, when you started to day 3, it's an order of a couple of megabytes, nothing big. So sample preparation protocols are usually some sort of Excel files, which are not really big. It gets an exponential growth from day 3 to day 8, and there is contribution from a sample pre-processing there. And it plateaus because sample interpretation is, again, doesn't add much. It's just reformatting what you already have detected during your processing and putting into content. But that was in 2007. That's what happened in 2012. What happened actually is obvious from day 3 to day 8. This is a raw data collection and processing. Instruments start collecting data much more aggressively, much more rapidly. So, and that's where we now. So we are plateauing that average project, and this is not a typical study for screening, but it's a typical clinical study that takes the pictures here. About 1 terabyte in size when you put it all together. That's a lot of data. So what's in that metabolomics data set, which will be deposited in? So collection of samples, typically basically 6 groups of animals, 6 samples. So each of those samples that's produced into these animal studies. For each typical sample, today we collect 4 different LC-MS injections. Why 4? We want to be comprehensive in our targeted endeavor because there are available 2 different chromatographies, one reverse trace, one fillet, and there are 2 polarities, positive and negative. And although there are some redundancy of what we are measuring in between, it's not as overwhelming redundancy, about 30% of components, that's all we have in all the labs in between those methods. So it's worth time-wise to measure all 4 injections. Then we try to record, but it's not always possible at the time of experiment to know your data origins. There are also possible some blind or even double-blind studies. You just receive samples, and all you know is a barcode. That's all you know. Later in the game, they will give you an whole explanation, but you don't. Then sample grouping, you're absolutely needed for analysis stage. If you don't know how to group your samples, you cannot drive any statistics. Obviously, I already talked about sample preparation record. And then there's a methodology for data collection. So you want to know exact parameters how you collect data for the state if you ever go back and you want to reproduce it. And methodology is not the same for every type of sample. It's different. It depends what you're targeting, what type of molecules you're targeting in your sample. And obviously, there is a collected data, and I'll talk about those topics. So let's look at what we want to capture. And this is more like a philosophical question. I didn't limit our project and even try to address all of it. It's just out there for the audience to think about. So what goes into as a sample of origin? So we want to know what type of biological species we are dealing with, which compartment we are assessing. So sample might come from blood, it might come down the stream when organism is eliminating something. It could be urine or fecal extract. It could come from your brain, or it could be a tissue homogeneous, some sort if you are doing specific programs. So sample preparation records, we talked about this very quickly. This is when I showed first and we started exploring what's inside there. I was thinking the student was going to quit on me because we dumped it to a text file and there were several pages of different instrument settings on top with the calibration record and many other things. But fortunately, Alexa saved me. It told me a joke about programmer and light bulb. You know those jokes, right? So I told him, don't worry, Joseph, this is vendor's problem. So this is my picture of what we currently collecting in vendor format. So there's tune methods, instrument methods, nicely preserved for our sample information. But what's most important there is data, and there's lots of it. What goes into data, and this is where Allostrop wasn't prepared for that, and I'll show why. So each point which we are collecting, and even the quiet region is full of signal. So each point has associated at least 6 double, and it's 16-bit variable, so lots and lots of data. And in my estimate, there is per 18 minutes run, there's 40 million points in today's rate. So here's our block scheme of first version of raw to ADF converter. And there are lots of technical details, but mostly we try to follow the same KISS principle, which we talked previous 2 days before. So anything we can readily match to ontology, we convert it and recode it. What we cannot match, we call it unknown and preserved in a special file so we can match it in the future. Now, question of how do we model this humongous array of mass to charge ratios and different retention times. And this is how our first implementation, and source code I think would be available. We will deposit it on our shop site. So model as one scan in one data cube and try to put some conversions, but that process is very, very slow. So here's some example of how I would say not efficiency, inefficiency. So starting raw file size about 153 megabytes, output ADF file size is with a compression about 800 megabytes, and it took about 40 hours to do that. And obviously, here's some technical implementation details. So now there are some questions over there. So obviously, one-to-one conversion is just the first step in this endeavor, but what we really want to think about how better organize our samples. Maybe we can pack more into it. And there are some other options. So depending whom you are talking about to on our team, so biologists who looking at a system biology level, they want it whole project in one ADF file, but I don't think it's technically feasible right now because of the project size. And here are some other ideas. We have pre-standardized preprocessing algorithms, so we're studying with that many signals, but we can reliably convert them into maybe some metabolites, so maybe ADF can store an algorithm which can be future propagated and everybody can apply to it. And with that, this is kind of a quick description of the data we will be posing as a challenge to the community. And those data were published, and I would like to finish with acknowledgement and show you some view of a beautiful Lawrenceville site. Thank you.